Okay, that seems to be working. All right. So first of all, let's see. Yes, so thank you, Weihao, for agreeing to giving a talk. You mentioned in our email exchange that your work is a bit more computer science than perhaps we are used to at the department, which is why this talk will be particularly interesting for us, as we'll get to see something different from kind of what we usually see. So thank you again for still agreeing to giving this talk. So Weihao will be talking about visual decoding from reversing human visual system. And without further ado, I'll let you take it away. And yeah, go ahead.
Hi, thank you. Thank you for organizing so amazing seminar. Okay. I'm Weihao. I'm said, I'm a student from computer science. So today I would like to introduce our recent work called Jing. My supervisor is Jing-Hao Xia and Xiaochen. And this work is a collaboration with INRIA and also the University of Cambridge. So yeah, I will get started. So in this work, we present Dream, a novel method for visual decoding of brain activities, which relies on reverse human visual system. So what is visual decoding first? So when you're using an FR machine, the brain activity is caused by the visual stimuli here, as you can see the animal, so can be recorded. The task of visual decoding consists into reconstructing the viewed image using the brain responses, which are usually encoded as voxels. So solving such tasks would not only enhance our understanding of the human visual system, but also will establish the bridges between human vision and also the computer vision. However, the limited availability of fMRI data and the complex nature of brain activities still present significant challenges to obtain fine-grained and current visual decoding results. Recently, based on the pre-trained different models, these recent visual decoding methods has shown to significantly improve the accuracy of some high-level semantics, meaning that a coarse description of the image will be reconstructed. But still, these methods here the results from a brain diffuser reconstruct images that suffer from important and color spectral inconsistencies. For example, the leftmost examples show how the reconstructed image described a cowherd next to a forest, but fail at inferring correct spectral information and color tunes. The same happens in other examples. So to mitigate the loss of color and spectral information, our proposed method called Dream addresses visual decoding based on the principles of human visual system. Specifically, we introduce a novel formulation based on the fundamental knowledge of the human perception that aims to reverse the pathways specialized in extracting semantics, depths, and color cues directly from the fMRI data, therefore mirroring the forward process within the human visual system. As a result, our method Dream outperforms state-of-the-art methods in visual decoding. Here are the overall pipeline of our proposed method. On the left is the overall descriptions of the forward pathways in the human visual system. So when humans perceive the visual world, the retina in the eyes capture incoming photons and converts them into electrical and chemical signals, which are then relayed to the brain, resulting in visual perception. Schematically, color and depth information are processed through the parvocellular and also the magnocellular pathways. The deciphered information are then channeled into the visual association cortex, abbreviated as the VAC, for retrieving high-level semantic cues of the visual images. So in our method, we aim to reverse the processes within the human visual system. Therefore, to deciphering concurrently the semantics, depths, and color cues from the fMRI recordings. So as illustrated on the light subfigure. So considering the fMRI data as the input, DREAM uses three components. The first component called the reverse VAC pathways. It will extract semantics in the form of the clip embedding from a large trained neural language model following the literature. In addition, we also propose the reverse PKM pathways, which deciphers color and depth in the form of called spectral palettes and also the depth maps as shown here. So finally, the reconstructed image will be obtained by feeding the obtained semantics and also depth and color informations into a pre-trained T2I adapter in conjunction with stable diffusers. So our first module is called the reverse VAC module. So the role of this module is analogous to that of the VAC component in the human visual system. So in other words, it is designed to extract semantics. It is designed to extract semantics from the fMRI data. So to do this, we train our encoder to align fMRI features from our brain fMRI encoder with the pre-trained and fixed clip space by using constructive learning. So here is the first loss. We use the initial construction loss to align the fMRI embedding with both the clip image embedding and also the clip text embedding. So given the limited availability of the fMRI data, so we also introduce a data augmentation strategy called Mixco to address the imbalance of the data between original clip training samples and also the limited availability of the fMRI data. This one will generate mixed fMRI data from convex combination of two fMRI data samples. With this allows extracting semantics while this allows extracting semantics, but this knowledge is inherently bounded by the clip space capacities itself. So, which means this alone cannot be encode the spectral color and the geometry. So to address this issue, we propose the second module called the reverse PKM component. Its goal is to invert the forward pathways of the parvo, corneal, and also the magnocellular pathways, therefore enabling the predictions of the color and depth from fMRI data. So we approach this task as an RGBD estimation problem, which means we want to predict color images and also depth images from the fMRI recordings. And then this color palette can then be obtained from the RGB predictions by first downscaling it and then upscaling it back to the original size. So to combat the data scarcity issue of the fMRI modality, we introduce a multi-stage encoder and decoder training strategy. So in the first stage, we train an encoder to map the RGBD to the corresponding fMRI data to a given the limited pairs of fMRI and the image with depth maps. So in the second stage, we train a decoder to reconstruct the fMRI recording to the RGBD images. So in the third stage, we fine-tune the decoder using external natural images in self-supervised learning. So all this together allows us to learn an efficient fMRI to RGBD mapping, therefore deciphering color and depth cues from the fMRI signals. So given the deciphered cues of the semantics, depth, and color, we now aims to reconstruct the viewed image from the three guidance. So we use here the powerful stable diffusion, also with a pre-trained T2I adapter model to inject the semantics, depth, and color information as the guidance for the of the generative process. So the result image will the final resulting image will be the output of our dream method. So to evaluate the performance of our method, we show reconstruction results of our method in comparison to the most recent baselines. So for the sake of comparisons, we show left the test image seen by the subject. So as shown, our proposed dream decodes the visual stimuli from fMRI much better than all the other baselines, with better spectral and also color arrangement, but also better semantics. So for example, on the leftmost, we show how dream dishes is like both it's both more occurrent visually and also the one perceiving the spoon here. So on the right, so our method, the reconstruction from our method, so it's the only one that recover the three players on the baseball field. So the same performance boost is also observed, can be observed in the table. So showing that our method perform better on the high-level metrics and also on bar or better with low-level metrics. So on the last bottom rows, so we show that each of our components contribute to the best performance. For two examples, we report details, visual results showing the variations of our reconstruction results. So on the depth and color predictions are in line with the pseudo ground truth despite the extreme complexity of the task. So at the results, we know that dream reconstruction close match to the test image. For comparison, we also deactivate the color guidance in the last three rows, which highlights the benefits of introducing such color guidance. So as you can see, the color consistency is much better if you introduce the color guidance for the final reconstruction. Very interestingly, while faithfully recovering the original depth from fMRI is nearly impossible due to the information loss in the capturing process of the brain activities. We know that the coarse depth is sufficient enough to guide scene structure and object positions, such as the orientation, such as the location and also the orientation of the animal. Likewise, the coarse color predicted from the fMRI recordings also provide reliable construct and guidance on the overall scene appearance. We also measure the benefit of our predicted semantics, depth, and color in the bottom table, which align with the visual judgment and observations. So here are more results of our decoding results. More examples of our visual decoding results. It can be seen that our method can produce consistent results in terms of semantics, appearance, and also the geometry. So one of our contribution is to introduce the color guidance. So we also evaluated the spectral and color qualities of the reconstruction results. So we borrow some metrics from depth estimation and also implement some color consistency metrics by ourselves. So as shown here, our method consistently outperformed the very recent methods, brain diffuse and also the Mandai method baselines. Yeah, basically that's all of my today's representation. So more details on the method evaluation and ability studies can be found in our paper. So you can scan the QR code to our project page. Thank you.
Hi, Weihao. Thank you for that. So we have plenty of time for questions. That was very interesting talk. And as I was mentioning earlier, it's always very nice and refreshing to see topics that are either hybrids between statistics and another field, or just applications which are really diverse compared to what we normally get to see. So thank you for that. All right, so do we have any questions online or in the room that we can start with? Yep.
Weihao, can you hear me?
Yeah.
So I was wondering how easy or challenging it would be to extend this. So I think at the moment you're just considering a still image. And I was wondering how easy or challenging it'd be to look at a video, for example, or a sequence of connected images. And whether you could, like, sort of, whether it would be possible to add a sort of time series structure.
I'm sorry, I can't hear you very clearly. Could you please maybe to the using the Mac or what?
Yeah, you might have to get closer. Otherwise, I could repeat the question. Okay, I'll just repeat the question for you, Weihao. So Luke was asking, currently what you're doing with your method is with a still image. And he was wondering how easy or complex it would be to do it with a sequence of images or a video.
Yeah, absolutely. Actually, there is already a sublimation working on decoding radios from the brain recordings of the brain responses. There is actually indeed, yeah.
Is that quite different from the method that you're using?
I think it's because most recent methods on neural decoding is just relies heavily on some pre-trained models. So it's so as I can remember, that method tried to decoding radios from the brain signals using the same different models that we use, and just adding some constraints on the in the temporal level. So in one word, I think it's possible to reconstruct a radio based on our method. Yes.
All right. Thank you. Any more questions from the room or online? So in the meantime, I guess I can, oh, yeah, Baptiste, go ahead.
Hi, Weihao. Thanks for the talk. I was wondering one thing, not very statistical or computer science, but practically how were the MRI data collected? So they need to go in an MRI scan to. I was not sure how the image are seen by the people.
Yeah.
Yeah, tell me more.
Oh yeah. The data we use is from a large scale fMRI and image pairs. So it's collected by using fMRI machine. So at each test, a subject is asked to stare at an image like a few seconds, and then the fMRI machine will record the brain responses.
Okay. So it was done on a specific machines maybe, and they look at a still image. It's not a real life looking. So for the moment is they look at an image and
yes,
and you've got the data. Okay, that makes sense. Thank you.
Yeah. So neural decoding is to reconstruct the viewed image from the brain recordings, not like capture the brain responses and then convert into an image. Not like that. Yeah.
And if while I'm speaking, if I can ask another question, how it seems quite critical to reconstruct the image. I guess it comes from a bank of image. All these reconstructed images, they must come from online or banks of image.
Yeah, it's from collecting large scale images is not that easy. So they directly borrow an existing visual image dataset called COCO. I'm not sure if you know that. So they select the image from an existing image, large scale image dataset, and then
Okay.
yeah.
Okay, yeah, no, I was not aware of that, because I guess that's quite critical for the quality of the reconstructions.
Yeah.
The more data you will have, the more image you will have, the closer you may get back to the truth. No, yeah, that was interesting. Thank you.
Thank you.
I have a question for you, Weihao, in the meantime that we wait for more questions, either from online or the room. So in one of your previous slides, I believe it was the one where you explained the reverse PKM.
Yeah.
So you had at the bottom, yes, this one. So you had at the bottom right of this slide the kind of a summary of how the data mapping works, and you explained that you go first from the image to the RGBD mapping, then from the RGBD mapping back to the image, and I didn't quite understand what the last stage was. I understood that it's a refinement step for parameter tuning probably, but I'm not it's not quite clear to me what those double arrows are doing.
Yeah, so sorry for making that not very clear. So actually the first stage, this one is actually the fMRI recordings. So in the first stage we want to, so we have images, and then we use some existing depth map estimation method to get the depths, so called midas. So it's like we have a data generation or preparation process to get the depth RGBD samples. So we use some pseudo ground truth of depth. So actually this, because we want the image and also depth as the output, and we want want to get the RGBD both from the fMRI recording. So we have to get the RGBD as the ground truth first. So in the first stage, so we have a data preparation to get such training examples. And in the first stage, it's actually not from RGB to RGBD, it's from RGBD to fMRI. So in the second stage, we train the fMRI to the RGBD. So because the training data is not very sufficient, so we also use like so that's why we introduce the third stage. We want to introduce more external natural images to guide the training process. So because the final goal actually is to predict the RGBD from fMRI, so we do not care much about the encoding process. So we actually, in the third stage, the encoding process is fixed, and we only fine-tune the decoder by using the RGBD pairs.
Okay, I see. All right. Yeah, thank you for that. I was missing that fMRI intermediate step. And I have actually a follow-up question with that. It's probably a silly question, but I'm just not familiar with this type of data at all. What does the fMRI data look like?
It's in the fMRI data from the fMRI machine is actually a voxel, but in our experiment, it's just it's more like it's just a one-dimensional signals. It's more like a voice or some time theory time sequence data. It's more like that.
Oh, okay. Okay, I see. All right. So I guess we could see if we have any more questions in the room or in the chat or online. All right. We have another question from the chat. I don't know if you can see that, Weihao.
Could you please?
Yeah, of course, because you're screen sharing. So it says, I was wondering if this method could also be combined with self-supervised methods, and that's from Iman.
Yeah, of course, because I think this is a very good question. Yeah, because the self-supervised learning is very useful when there is not enough pairs for training. So we and in our case, so collecting fMRI recordings is actually very hard to acquire. So that's why we introduced the third stage training. So the fMRI data is like this examples. It's more like a few thousand. So I think it's not enough to train a very large scale model. So that's why we introduce a third stage that we can use image and depth only without pairing the fMRI recordings. Yeah.
Does that answer your question, Iman? All right. Okay, well then, in that case, I think we can wrap up today's session. Thank you again, Weihao, for this short talk on your work.
Thank you so much for you and Luke to organize this seminar.